So yeah, thanks for the kind introduction and the opportunity to share some of our work with you here today. The work I'll be sharing is mostly in this area of probing B cells, asking the question, what are sort of optimal design rules at the molecular scale for vaccines, where the cell-based work was done in Daryl Irvin's lab. It's been a close collaboration with Daryl's group over the past couple of years. At a high level, my lab in biological engineering is working on several different areas where we use programmed nucleic acids. Programming nucleic acids actually dates back to the Department of Biology. Ned Seaman, who is sort of the founder of the field, was a postdoc with Alexander Rich in the 70s, working on crystallography, and he had the idea of using nucleic acids as programmable materials at the nanometer scale, and he started to explore many applications using this technology and approach, and our lab is still doing that today. I'll be focused on its applications to some very early studies that we're trying to pilot in the direction of vaccines and therapeutics. Everyone's familiar with the sort of overarching motivation, which is that there's a real pressing need in the coming decades for new gene therapeutic, in vivo delivery in particular, but vaccine technologies as well. We have now identified over 6,000 genetic disorders, thanks to next generation sequencing and large scale efforts across the globe, really, in that direction. Centered at the Broad Institute as well. In 2019, there were over 500,000 deaths just alone in the US from cancer. And to date, remarkably, there's been already over 500,000 deaths, as everyone's familiar with, from COVID-19, the disease caused by SARS-CoV-2. And then finally, a very important other pandemic that's been ongoing for decades now without either a cure or a vaccine is AIDS, caused by HIV-1. Clearly, of course, centered on the developing world more than the US today in terms of its ravaging impact, but nevertheless, exorbitantly important for the development of vaccines as well as cures. So in our group, we've been exploring and continue to explore a very early stage nanotechnology for diversity of applications across these disease indications and across multiple different types of modalities of therapeutics, mostly targeted towards the gene therapeutic space. And everyone's familiar with this sort of rise of siRNAs and ASOs and messenger RNAs, as well as now CRISPR, of course, as next generation therapeutic tools for curing or treating a variety of diseases, from cancer to genetic diseases. The challenge in this space, or one challenge, there's many challenges, but one challenge has been targeted in vivo delivery of these genetic tools and therapeutics to specific organs of interest, cell subtypes, and then of course having safety and toxicity concerns satisfied, as well as high efficiency of transduction of either gene knockdown or expression. Actually, just last week, published a sort of review article in this direction with Marina Dobrovolskaya, who's the head of immunology toxicology at the Nanotechnology Characterization Lab, who's been working on these nucleic acid materials for some time now, talking about some of the challenges and opportunities. These are just sort of schematic therapeutic constructs that I have on the left, where you're looking at, I should have gotten my pointer. See if I can do that real quick so that I have the spotlight. So in one case, GP120, the outer envelope protein of HIV, is scaffolded in 30 copies around an icosahedral 25 nanometer particle in sort of a vaccine type application. In another couple examples, a messenger RNA is either complexed on the interior of a small nanoparticle made of nucleic acids in its sort of native folded state, or it's alternatively hybridized to oligos that make it form this discrete particulate shape. where it's not ready for translation per se, but these are hybridized to, the messenger RNA in red is hybridized to DNA oligos that, if subject to degradation through DNase 1 or 2, would be ready for translation potentially. And then in the bottom, you're looking at CRISPR and sRNA payloads, where the sRNAs can be hybridized to the inside of these particles. Dan Anderson had a very nice Nature Nanotech paper on this several years ago, showing how that could be done. And then in this case, we also have a ribonuclear protein complex with guide RNAs of the CRISPR RNP hybridized as well inside of the particle. And so in each case, these particles are homogeneous in size. They're virus-like particles in terms of their size and geometry on the 10 to 100 nanometer scale. They're relatively inexpensive to manufacture. We can talk more about that. It always depends on what one is relating to. But compared to AAV, for example, it's inexpensive. It has capabilities because it's made with these synthetic oligos of active targeting using peptides or sugars or ligands, small molecules, and also immune cell stimulation. There are concepts of triggered cargo release, whether it's sRNAs being released or CRISPR RNPs in response to pH changes or other environmental cues, tissue-specific DNase 1s. And then they're biodegradable. So this could be a good thing or a bad thing, depending on how you view it. Some nanotechnologies are extremely stable and pose challenges for safety and toxicity in terms of demonstrating that they're metabolically processed and cleared from the body. In this case, these are made from nucleic acids and can be modified to be more or less stable, but in either case, they certainly do degrade under the action of DNase 1. So in our lab, we're technologists, not immunologists, and so we are developing these materials for a variety of indications. It's, again, early days. For these materials, we've had an ongoing wonderful collaboration with Daryl Irvin's lab on the HIV direction. And then more recently, because this is a platform technology, we've been able to pivot it towards SARS-CoV-2 by swapping out the HIV antigen of interest and swapping in the receptor binding domain from the spike protein together with Aaron Schmitz and Daniel Lingwood at the Raygun Institute. So as alluded to before, there's still no successful vaccine for HIV AIDS, despite decades of research. There are a number of reasons thought to be why this is such a challenging pathogen to develop a vaccine for, surrounding its mutagenicity, its glycosylation patterns, its inaccessibility of a conserved epitope for targeting antibody response, as well as a low and variable copy number of envelope proteins that it actually carries on it and expresses. In any case, in the mid-'80s, HIV was identified as the cause of AIDS. In 1998, large number of years later compared to what we're seeing today in terms of vaccine rollout, the first large-scale vaccine trial began. And then it was only in 2004 that candidates failed in phase 3. And 2010, quite recently, 10 years ago, that it was identified from patients living with HIV that had developed some broadly neutralizing antibodies called VRC01 and VRC02 that neutralized most strains of HIV. That these then were put into pipelines for drug development for antibody therapeutics, but also emerged as a new target for vaccines aimed at eliciting antibody response, VRC01 in particular. And I'll be talking about an antigen from the Scripps Research Institute that Bill Schieff's lab has engineered, that Daryl's lab has also worked on, that we're using in our nanoparticle formulations, and that's actually an ongoing clinical trial now for HIV vaccine. Very different timeline, and hopefully much higher expectations, although cautiously so, for the recent pandemic is the coronavirus vaccines that have emerged. So in this case, there are over 160 vaccines that are currently in development. Number seems to rise regularly, but there has been all over the news, including the very high profile and promising approach from Moderna locally in our neck of the woods. That's a messenger RNA vaccine. That's already entering phase 3 together with a few other candidates that have already passed phase 2. And by and large, these are looking to elicit antibodies to target the spike or spike protein of the coronavirus that mediates entry into human airway cells through the ACE2 receptor. So one might ask, why do we need another vaccine in this direction? Why is our lab exploring our platform to be applied to this direction? Well, I think that the fact that there are 160 vaccines speaks to the incredible technological capabilities that we now have in biotechnology on the one hand, but it also speaks simultaneously to the uncertainty and lack of any one or two sort of winning guarantee technologies that we know are going to work, that everyone just goes after and produces a successful vaccine. So there's a lot of uncertainty about the persistence of antibody response that might be elicited by these different vaccine technologies in efficacy that will need to be demonstrated over many months and years. And so there's uncertainty, and there's really need for taking as many shots on goal as possible, in the words of Gary Nabel, who formerly was head of NIH's Vaccine Institute. So there are a few different formulations that are in that group of 160, and this helps put our formulation into context. So they're the genetic vaccines that we mentioned with Moderna. This is either a messenger RNA or DNA that produces the antigenic proteins of interest. The RBD in particular is a major target. And examples are, in addition to Moderna, Translate Bio in our neighborhood, as well as CureVac over in Germany for RNA vaccines, messenger RNAs, and then Inovio for DNA-based vaccines. And these can be delivered with lipid nanoparticles, for example, or through sort of mechanical electroporation. The Oxford vaccine is also now in phase 3. That's just in the news also very recently, is a variant of the common cold, adenovirus. In this case, it's a viral particle that is genetically modified to express, for example, the RBD or receptor binding domain of SARS-CoV-2. Because it's an existing virus, has very, very good transduction capabilities. Some questions about being able to boost this vaccine, given that there may be pre-existing antibodies or antibodies that may neutralize the vector. But in any case, it looks very promising so far. And then the final two categories are the ones that have been around sort of the longest and most successfully. They're either whole attenuated virus vaccines on the right, a weakened or inactivated version of the virus to provoke an immune response, and then protein subunit vaccines, which is the category that our approach falls into, where protein fragments together with various formulations from polysaccharides or just protein scaffolds are used to provoke immune responses. And so if you look historically, in any case, most FDA approved and licensed vaccines are of the last two categories, which are sort of the technologies that have been around a bit longer. Protein subunit or attenuated virus vaccines, and they elicit high antibody titers. There are sort of three branches of immunity. There's cell-based immunity in addition to innate immunity and humoral immunity. And this sort of longer list of vaccines mostly work through humoral immunity, producing antibodies or eliciting antibody production that is in memory for long periods of time. And we know of many vaccines that are very, very successful to keep us immune for decades or longer. There are two important ones here that I want to point out, which was for hepatitis B and HPV. They're virus-like particles. So these are self-assembled viral protein particles that look like the antigenic viral coat itself, but they're assembled from the antigenic protein itself of interest and are also therefore safe. They don't have, well, these are all approved and safe, of course, but in this case, they have seemingly higher safety, even because you don't have genetic material that might cause some concern anyways across the broader population. These virus-like particles are shown to traffic robustly to secondary lymph organs, lymph nodes in particular, like viruses. and are very stable when made from proteins where B cells are then activated. The sort of germline B cell pool that see these vaccines or virus-like particles goes through a set of stages of selection and what's called affinity maturation to hopefully elicit the antibodies, the neutralizing antibodies of interest. Importantly, these virus-like particles are first presented by dendritic cells in an intact antigenic form. So rather than being a single protein subunit, they're intact as a viral particle. BCR, the B cell receptor, then is robustly cross-linked by these, what on these virus-like particles are repetitive arrays of this antigen that cause strong immune cell stimulation at the level of first recognition activation and signaling that then the antigen goes through MHC2 processing to present peptide fragments to T cells that elicit help, and if selected, then the B cell will migrate to the dark zone of the germinal center, where it will be basically undergoing somatic mutations in this antibody to produce daughter cells that will either apoptose if the affinity of the antibody is not improved for the antigen of interest, and otherwise they'll be selected in a positive sense and either selected to go forward for subsequent rounds of somatic mutation and affinity maturation as one generates these antibodies to have high affinity and specificity for the antigen of interest, or exit and become plasma cells that might migrate to bone marrow and be present for decades, producing antibodies of interest for neutralizing the antigen, or memory B cells that can later respond quickly to new infections. So that's sort of an overview of the B cell maturation process and how the vaccines interact with them. On the virus-like protein direction, some of the earliest work of engineering these using sort of nanotechnology approaches is from Gary Nabel, who took ferritin, a human protein that assembles into a 24 copy number nanoparticle cage. You can see the size is sort of 20-ish nanometers or even smaller. By fusing this genetically to HA, which is an antigenic protein from influenza, H1N1, hemagglutinin, one is able to basically self-assemble this particle to have 24 discrete copies of HA expressed, and you can, on the surface of the particle in a virus-like antigenic form, where you have these many copies that should induce a strong B cell activation and triggering. And you can see from structure TEM that one gets high resolution and fidelity of these virus-like particles. They also showed in vivo very robust immune response and antibody titers. This technological application in only 2013 has evolved quickly thanks to Neil King and David Baker at University of Washington. They've done a lot of work in the past few years to extend this approach of making protein-based nanoparticles to present a variety of antigens, and including SARS-CoV-2's RBD most recently. These particles have many advantages, including their stability and expression capabilities in terms of producing large quantities inexpensively. A few limitations they have is, one, they have a limited but pretty broad control over antigen copy number, spacings between antigens, as well as the number of antigen types. They, like adenovirus, might produce neutralizing antibodies if one needed to use multiple boosts or multiple injections. One might have neutralizing antibodies that would bring them out of commission. And then finally, their glycosylation patterns, which can be very important to vaccines such as for HIV, may be non-native. Another variant that brings us to the focus of today's talk more is from, as I mentioned, William Sheaf's lab at Scripps Research Institute. So Bill made a bacterially assembling or produced in a self-assembling protein-based particle, a virus-like particle. That's a 60 mer from the engineered outer domain of GP120, which is the envelope protein or trimer from HIV-1. That's the major target for vaccines and also broadly neutralizing antibodies. Importantly, he engineered this to be germline targeting, as well as to target VRCO1 class B cells. And the reason that was important is because the VRCO1 germline B cells don't actually bind or respond to ENV in terms of their affinity, the envelope protein. And so the idea was to use this as a priming candidate to bring B cells to a point where they would then be able to be boosted by ENV to produce these VRCO1 class antibodies. These bind the yellow domain shown here, which is the CD4 antigenic region on the envelope coat protein. So that's actually in clinical trials now. There's a single vaccine construct that was studied in vitro and in vivo. In our work, we were interested in understanding what the design rules are for these vaccines, in particular asking the question, how does the copy number of antigens affect B cell triggering? How does the spacing between these antigens affect B cell triggering and also antigen affinity? And how does scaffold rigidity also affect B cell triggering? These are all questions that are of great interest for general design rules for vaccines and B cell recognition of virus-like particles. And a variety of different technologies, from lipid nanoparticles to protein scaffolds to polymers, can sort of target one or two of these questions, but not all of them. And with DNA origami or DNA nanotechnology, that again, I mentioned dates back to really Ned Seaman, we're able to answer these different questions in vitro so far about what these optimal design rules are for B cell triggering using this antigen and a few variants that I'll share with you. So this technology that Ned really published in a theoretical seminal paper in 1982 postulated that once you have synthetic DNA, you can use Watson-Crick base pairing and hybridization to bring different regions of DNA together to hybridize, that we do all the time, of course, in hybridization in molecular biology. But he also did something more subtle, which was to co-opt holiday junctions that are commonly used in recombination or repair, and recognize that he could, with synthetic DNA, program these crossovers, these double crossovers, to be present to cross-link neighboring duplexes of DNA that then would bring them into proximity with each other. And he showed that you could make custom crystalline patterns to host guest molecules, for example, using these simple strategies. Paul Rothermund in 2006 then invented this term and technology that took the rules from Ned Seaman about how to make these materials, but he brought it to a sort of virus-like dimension and scale by using a long single-stranded DNA scaffold that you'll see on the right. In the field's case, mostly comes from M13. The genome of M13 is around 7,000 bases. It's a phage that infects E. coli and has a known sequence, a fixed sequence. And if you design oligos to be complementary in Watson-Crick base pairing to that M13 sequence, then you kind of fold that scaffold up into a target shape as you cool the solution from high sort of 90 degrees C down to room temperature or physiological temperature, and you form that final shape. And these oligos are also called staples, and it's called origami because it's much like taking a piece of paper and folding it into a swan, for example, and then stapling the fold so that it forms that final shape. In this case, the stapling is done by all of those oligos, and the folding is not paper, it's this single strand of DNA that's from M13. William Shee's lab also produced three-dimensional brick-like objects from this technology. And then our lab in 2016 introduced a computational approach to design from the top down, meaning taking a simple geometric 3D shape and producing the sequences needed to fold that target shape. into any virus-like particle geometry on the 10 to 100 nanometer scale, using either 2 duplexes on an edge or later also 6 duplexes, which changes sort of the rigidity of the particle and the size. But one has access to a broad space of geometries. And because the oligos are synthetic, they can be produced with overhangs also that can then be targeted using simple hybridization to hybridize an siRNA or PNA, peptide nucleic acid, with a cargo of interest that's a therapeutic or a targeting ligand. And this is just showing sort of the routing of the geometry, the scaffold, and the oligos. And then we also have in our lab also some click chemistry approaches that allows us to click on ligands and small molecules as well. And now currently generate sort of large scale libraries of these particles that we're using for various delivery applications. The production of these at preclinical scale is something now that's possible where there are a couple approaches, but we're not dependent on the M13 7000 base native sequence of that phage, but rather use a helper plasmid from Andrew Bradbury, who used this originally for phage display and antibody engineering, but we use it for producing custom sequence and length scaffold of DNA, and there are a few other published approaches, but we can now make single-stranded DNA in fermenters that allows for large scale, low cost production. So this is no longer a rate limiting step. We still do use phosphoramidite or solid phase synthesis for the oligos, which is the same technology that is used in siRNA therapeutics, for example, production and manufacturing. Once one makes these materials, one can see that you get a very nice sort of folding of single particles shown here on the sort of 20 nanometer scale. These are all individual pentagonal bipyramid DNA origamis. One can also look at high resolution 3D cryo-EM and see that this icosahedron is also the shape and size with the duplexes of DNA running through it of interest. And then this then forms sort of the scaffold or breadboard on which we can position antigens. And so in this case, Daryl's group took the EODGT8 antigen that was this engineered outer domain of GP120 from Bill Sheep's group that they've been working with, and added a peptide nucleic acid 10-mer that hybridized to a single-stranded DNA oligo that we presented in variable copy numbers on an icosahedron, as well as a few other particles, as well as a simple rod to ask just very basic questions about how distance between antigens impacts B-cell triggering. And from these different types of molecular designs of antigen presentation, we can carefully control the copy number of the yellow EODGT8 as well as its position in 3D and 1D. Which is challenging to do, if not impossible, with equivalent protein-based technologies. And so the question then is really how do these parameters affect B-cell recognition and triggering, and can we use that then in vivo later to improve our vaccine design? So the first observation was taking the icosahedral DNA nanoparticle that's around 25 nanometers or 30 nanometers, and just varying the copy number. What you're looking at is fluorescence on the vertical axis and time on the horizontal axis. This is looking in a fluorescence plate-based reader at B-cell fluorescence, where the B cells are a reporter line, a Ramos cell line, where only IgM for VRCO1, I should say, is expressed in a blank background. And so this germline targeting EODGT8, basically when it engages the IgM receptor, there's a fluorescence pulse due to calcium signaling that occurs. And so he developed this to be used specifically for screening in vitro different vaccine candidates. And what you can see is on the baseline, when there's only one copy of the antigen or none on the particle, the DNA nanoparticle, there's no response, as you would expect and hope. And then the positive control is the EODGT8 60-mer with 60 copies. That's the protein nanoparticle. You see it's a red, very high response. The antigen is added here at sort of 60 seconds, and then the triggering is the rise in fluorescence. And the surprising result to us initially was that we found that we only needed 5 copies of the EODGT8 to trigger a similar level of activation in response as the 60-mer. And so you can see a monotonic increase in activation as one gets up to 5 or 10. And the other DNA nanoparticle, and not look at only copy number, but ask the question of how distance affects triggering of B cells. And in this case, we took the rod-like particle, and so the size of an antibody or IgM receptor on the cell surface is on the order of 10 to 15 nanometers. And if you're at sort of 7, we saw very low activation, and then a monotonic increase in activation as the distance between the antigens increased up to all the way to 30 nanometers, which is significantly larger than the size of a single receptor. And that persisted out to large distances, although we really need more data out at the sort of 100 nanometer scale to corroborate that large distance. But what was quite clear was that there's a monotonic increase in activation as we separated the antigens further away, which went a bit contrast to what one might expect, which is putting antigens as close as possible together. This result also bore similarly true in an icosahedral case where the lead author, Remy Veneziano, with Tyson Moyer from Daryl Irvin's lab, took 5 copies of the antigen and increased the spacing from 311 to 15 to 22 nanometers on the surface of the particle, and saw a similar sort of increase monotonically in B cell triggering activation out to sort of the 22 or so nanometers that they were able to go to. The other interesting question pertained to the flexibility of the scaffold. So antigens are sometimes presented on polymer scaffolds, and to ask the question, how does presenting a known distance or a given distance between antigens affect triggering compared to a flexible polymer crosslinker that has a similar distance measured in a sort of statistical sense as the distance between the ends of the particle or antigens in that polymer. And what we saw is basically it's a big difference that rigidity matters. And so, I think that as pathogens have evolved, whether they're have sort of a fluid-like membrane where the distances can change or be more fluid compared to fixed, might have implications also on B cells' abilities to potentially recognize them. We, in Daryl's lab, were able to also test this in a, not a dimeric form on a 1D rod, but in a 3-dimensional viral-like particle. So Eike Wamhoff in our lab with Ben Reed in Daryl's lab took a DNA-based viral-like particle and presented a peptide in this case that had a couple different affinities, a low and a medium affinity for BCR, and then they compared that to presenting the peptide on a liposomal formulation, so a lipid nanoparticle-like formulation, with similar densities and copy number of antigen or spacing between them on average, but saw higher triggering significantly in early time points in any case with the structured rigid presentation. And on a lipid nanoparticle, you can imagine that the peptides can diffuse freely. So even if the average spacing is 10 nanometers, on average, they can be many different distances from 1 nanometer out to 10 or 20 or 30 compared to the fixed presentation on the DNA nanoparticle. So that'll be an interesting question to explore going forward for a whole host of different receptor ligand binding interactions that might mediate endocytic uptake and trafficking and recognition and cell activation and so forth, where presenting on these types of fixed distances and copy numbers might really might be important in some contexts. To validate the activation from the fluorescence calcium signaling, Matt Stone in our lab also did some confocal microscopy where he fixed B cells different time points after activation, so either 1 minute or 5 minutes or 30 minutes, for example, with the icosahedron with 30 copies of the antigen. And you're looking at, in red, the EOD-GT8 antigen itself, the protein antigen of interest, and then BCR stained in green, and then phalloidin that's staining actin. Filamentous actin is a polymer that self-assembles in the cortex of in the submembranous region of cells that is typically activated and self-assembled in endocytosis as well. And so after 1 minute, you can see punctate forming where the vaccine construct is purportedly bound to BCR, and an actin shell also already starting to self-assemble. And then later you can see endocytic uptake beginning and internalization, which we have not yet characterized downstream signaling beyond looking at the very early signaling events that involve a couple of kinases, SYK and LIN, and confirmed that the icosahedron 30mer has a similar level of signaling concomitant with the positive control EODGT8 60mer, which corroborates some of the previous results, at least on the B cell activation. So one of the earliest models that we've come across that painted a picture of how B cells recognize antigens and get triggered is from Dinsis et al. In 1976, where they were able to use a polymer with an antigen that they were able to control the spacing of the antigen on the polymer, and did immunizations in vivo and looked at antibody production downstream, and came up with a concept of an immunon, which sort of lingers today and remains. And I should say that the mechanisms of B cell activation at the single receptor and cell level is an enormous field in its own right, with many, many, many dozens of papers that our lab and this study don't really address, and is really an active area of research. But what this early model showed us, or at least posited, was that you needed a critical copy number of these immunogens in a multivalent presentation in order to effectively form this immunon, which is maybe a little bit like cross-linking up to form this discrete cluster from which then B cells would be triggered and activated. And a single monomer wouldn't do that. And if you had too few copies, or if they're too far spaced apart, you wouldn't do that. And so they kind of came up with this critical copy number concept, as well as spacing using polymers. Our observations build on this. Our data suggests that the activation increases monotonically with the number of immunogens, which is different, and that there's something around 25 to 30 nanometer distance that's important, where we see this plateau, although again, that's relatively early. We need more data to corroborate that. And the fixed rigid spacing between the immunogens is also important for robust B cell activation, presented on these scaffolds, which either protein virus-like particles or DNA virus-like particles do. As a first sort of relatively simple explanation and mechanistic model of our results taken together, Jayajit Das, a faculty at Ohio State University, came up with a computational signaling-like model where he treats individual BCRs in the membrane together with downstream kinases, LIN and SYK, in a reaction diffusion type of computational model, and has done quite some work on this. He's actually from Arun Chakraborty's lab as a postdoc here at MIT. And in this model, he basically posited that it was a steric occlusion between what are microclusters of BCRs and signaling complexes, that as you bring those microclusters apart, or you trigger with antigen microclusters that are further separated, you have higher levels overall copy numbers of BCRs and signaling complexes that can participate in the signaling events and activation. And that's where this monotonic increase in activation comes from as one increases the distance between the antigens. And that's shown on the right in the distance D increasing between antigens in a pentamer or a dimer, with a relatively monotonic increase. And this is on the right, just showing the fraction of overlap in these clusters, which as you have less and less overlap, you have, again, more and more copy numbers and engagement. in the signaling process. So because these DNA virus-like particles are a platform technology, like many vaccine technologies are platform technologies, it's something that is very amenable to swapping out, for example, antigens. So in this case, in this variant application with Aaron Schmidt and Daniel Lingwood at the Reagan Institute, we were able to take the receptor binding domain from SARS-CoV-2 that was modified by graduate students Jared and Blake and his group, who did that in a matter of a couple of weeks, to introduce a terminal modification that allowed us to conjugate this to DNA virus-like particles produced in our lab in a matter of a few weeks, where we swapped out basically the HIV-1 engineered outer domain antigen for this RBD, and produced particles. And interesting questions here pertain to how copy number, again, of this antigen affect B cell recognition and triggering, and also presenting them in trimers versus monomers, as they are in the native versus non-native engineered constructs, how these impact B cell signaling. So it's still early days for those studies, but we're just getting ready to go in vivo imminently. And what we have seen is that the icosahedral particle from DNA with 48 copies binds to hex cells that are expressing ACE2 specifically, and similarly to wild type receptor binding domain on its own. In this case, the monomeric antigen does bind ACE2. It has high enough affinity. The particles bind similarly at either room or low temperature, and they don't bind also, of course, if the ACE2 is not expressed. That's the orange control wild type hex cells without any ACE2 expression, showing orange and no binding in each case. Yeah, so in both projects, we have a number of passivation techniques that, as I mentioned, these are prone to DNA degradation. They can last anywhere from a few hours to a day, depending on the modifications that are present in the DNA nucleotides, or PEG passivation to stabilize them. Very important questions for vaccine applications. So in the EODGT8 protein particle that Daryl's lab has seen resident in lymph nodes for weeks as a stable construct, and follicular dendritic cells also somehow stabilize particles as they recycle them through endocytosis, but don't actually degrade them, so that they can repeatedly present them to B cells in generating a humoral response. And in both Daryl's lab and also in the SARS project, we have a lot of interesting work to do to see how these results for molecular design rules might or might not translate in vivo, including trafficking to lymph nodes, very importantly, of course, as I mentioned, and stability, as well as eliciting T cell help, and humoral response. So one just final slide or two is a very important difference between our vaccine particles, virus-like particles, and the ones from Neil King and David Baker is that ours are made from DNA plus antigen, which is the antigen of interest for humoral response, whereas the protein particles from David Baker and Neil King and others, as well as Bill Sheaf, are protein scaffolds with a lot of protein and then some smaller antigenic regions. And I think a real focal point question for us will be, given that DNA is recognized by the innate sort of signaling and recognition pathways, either through endocytosis and endosomes, through TLR9, or cytozolically through CGAS and STING, that elicit type 1 interferons, how this downstream innate immune signaling in B cells, together with recognition of the antigen at the cell surface and then processing through MHC2, affects the overall humoral response. And that's something that Daryl and Daniel and Aaron are extremely well suited to study as immunologists, and we look forward to working with them on an ongoing basis to reveal some of these differences. So that's basically what I had to talk about today. So I hope I shared with you that DNA virus-like particles offer a versatile, rapid, candidate vaccine platform very early compared to many of the technologies we've seen, of course. For HIV-1, SARS-CoV-2, one can also present peptides as well as antigens and mixtures of 2 or 3 or more copies of different antigens to get broader representation of pathogens and even multiple types of pathogens. We are on an ongoing basis translating these in vivo, as I mentioned, with very important questions about stability, TLR9 signaling, and T-cell help. And we also have early studies that I didn't talk about today due to lack of time and sort of focus that are ongoing, where we're formulating these virus-like particles together with sRNAs and ASOs and messenger RNAs and CRISPR, which are in each of their own rights huge areas of research and lots of work to do for the treatment of cancer as well as potentially genetic disorders going forward, as we explore these tools for new as a new delivery tool for these therapeutic modalities in vivo. So with that, thanks again to the Marble Center for hosting me for the presentation today, for the first-rate postdocs and students who did the work that I shared with you. Remy Veneziano, who led much of the work on the HIV-1 project, has his own lab at George Mason University now, and Eike Wamhoff has taken over and done great work in that direction, together with Grant, who's a chemical engineer, first-year student, who's developing innovative strategies for attaching antigens of many different types to these particles and making large-scale combinatorial libraries. And then thanks to a great set of collaborators in this direction, and of course our funding sources. And I'd be happy to take any questions if there are any in the last 5-10 minutes that we have.